Hello everyone, and a very warm welcome to Soho Insider, and it's my true pleasure and privilege actually to moderate a discussion today with three wonderful colleagues. So as we start this discussion, I wanted to first and foremost focus on CELMoD mechanism of action, and so if I may, Katja, as you think of CELMoD mechanism of action and overcoming drug resistance, what comes to mind as what you think are the distinguishing features of this novel class of agent?
Yeah, thank you. I think it's a real innovation we see now with the CELMoDs. We are coming now from 2 decades of treating with IMiDs, immunomodulating agents, starting with thalidomide, and then turning over to lenalidomide, pomalidomide, and I think that truly changed the life of many myeloma patients. It enabled continuous treatment, and it enabled combination treatment, and it facilitated the whole immunotherapy emerging spectrum we now have. But on the other hand, it was also time to move into the next generation of those agents with now a more distinct targeting, and so we can really look on better efficacy and a much lower off-target toxicity, and I think that is the major feature of the CELMoDs.
Yeah, I would agree with you. I think it's important for our audience to share that they are quite distinct as a class from the original IMiDs, which obviously have transformed myeloma management over the last 3 decades. And I think, Thanos, as you think about what distinguishes CELMoDs from other degraders and molecular glues in the same space, how do you think of mezigdomide and iberdomide?
Yeah, I think that there is a more tight and permanent binding to cereblon. Which induces myeloma cell kill, but also specifically for mezigdomide, there are emerging data that this agent can modulate T cells and may have a role in the T cell exhaustion story, which is seen very frequently now that we have T cell redirecting therapies.
Yeah, so I think an important thing to understand is that this engagement of the cerebral E3 ligase complex involves closure of it, which in turn activates it and results in downstream transcription factor degradation. And so, Rahul, as you think of it, what strikes you as the most sort of remarkable feature of the CELMoDs, then, as you've come to understand its mechanism of action?
Absolutely. So I think in data that you've presented, the initial MESI-DEX paper that published in the New England Journal of Medicine, the responses are deep and they are durable, even in patients where lenalidomide and pomalidomide had stopped working, right? And I think 5 years ago, if you'd asked me, are IMiDs and CELMoDs, part of the same continuum, or are they different? You might have said, oh, they're kind of, the next generation of the same class. But here, they just work so durably, even when BCMA CAR T stopped working, even when Len and Pom have stopped working. The way I look at it is that, in table 1 of a future manuscript, of any other drug, I want to know what % of patients were exposed to Mezi or iberdomide, because that patient population is so different than just traditional Len or Pom exposure. In my mind, I think of them totally differently, just because of, again, how deep and durably they work. And that's on the myeloma cells. Thanos, your point about the T cells is really important, and I'm sure where the field will be headed as these drugs gain adoption.
So to build on that, Rox, because I think you make a very good point, is to say that in terms of cerebral E3 ligase closure, around 15 to 20% closure is what we see with immunomodulatory drugs. In contrast, iberdomide, it's actually 50%, and remarkably with mezigdomide, it's 100% closure. And I think these downstream effects are dramatic as we look at coefficients of degradation preclinically. But I think, Raul, you raise an excellent point about the activity in the clinic being what's so convincing. Correct. And I think we also see, Katja, don't we, remarkable extramedullary activity, which is not something, frankly, that we normally associate with the immunomodulatory drugs. And to see such dramatic effects of mezigdomide is quite remarkable. I think as we think about